Welcome everybody to our last seminar of July 2021 for the IMAG MSM working group in multi-scale modeling and viral pandemics. I really appreciate you coming. I know as the summer has been progressing, people have been disappearing for holidays and conferences. And so I appreciate the loyal following of people who are here today. We have two great talks scheduled. I need to remind everybody that this meeting is being live streamed on YouTube. It is recorded, and those recordings will be publicly available. I think nowadays Zoom tells you that when you log in, so you know it, but I want to remind you of that. As always, Reinhard and myself are available to take criticisms and suggestions. You have our email addresses. Jim Sluka and Bruce Shapiro are always on hand to help in every way possible. And we are always looking for your suggestions how to do better. We have our usual communication channels. Some of these get used quite a bit, some of them not so much. Please do help us update the IMAG MSM wiki page. There is an IMAG LinkedIn page. Please use it. Post your findings. It doesn't have to be something that was working group. If you have research related to the topic of the working group that you want to publicize on that IMAG working group LinkedIn page, please do it. It's a good opportunity. And my experience is that people notice that. And we have two actually YouTube channels where we have this material. Hold on one second. So if I can have people, if Bruce, if you can do a backup recording and Giuliano, that would be helpful too. So about that. Okay. So now we have a time for any quick announcements. Does anybody have anything they want to bring up quickly for this week? No? Okay, our schedule for next week is that on August 5th, we have Jim Sluka, who is going to present a draft white paper that he has been working on practical approaches to digital twins in medicine. Of course, this is a topic that we've been discussing a lot lately in a variety of ways. And we'll have him speak, and then we'll have an opportunity to have a group discussion on what our goals for our second year should be. We've been in business now for almost a year. It's a year since we started the discussion of creating the working group. And I think it's time to start thinking about what we could do better and how to go forward. Reinhard also, I think, feels strongly about this. And so anybody who has agenda items or suggestions for that discussion, please send them to us. These are things that we often discuss in the context of the steering committee, but we wanted to have those discussions in a more open forum as well. We still could use your suggestions for future speakers. We have actually a lot of people lined up for the fall, but there are some open slots. August 12th, we're going to have Enrique from Florida speaking again. TJ Sigo, who's one of our first speakers, is going to give another talk. But there are slots available after that. We have a lot of other people lined up. You can see the full lineup on our website and make suggestions. I know Dean Bottino from Takeda is planning on speaking. There's some other quite high-profile speakers who are lined up for the late summer and early fall. Please remember to mute your mic when you're not presenting. I'll give a 5-minute warning to our speakers. And with that, you know that I don't do elaborate introductions. I will turn the floor over to Gary Ann, who's going to talk to us about modeling the difference between human and bat immune systems.
Thanks, James. Let's see. let me see, should the presentation mode come up?
We're seeing it in your view, not in the presenter view.
Okay, let me try this.
No.
Is that better? No. Let's see. Use slideshow. There
Not a big deal one way or the other.
we go. How's that? That's
There
better?
you go.
Okay. All right. So as you guys can read this title, I'm a surgeon from the University of Vermont. I've been doing multi-skill modeling a lot. Most of you have heard me ramble on in this particular working group meeting. And what I'm going to do today is talk briefly about some work that we've done on trying to use a model to examine some of the differences between bats and humans. So this is a relevant issue because the most recent pandemic appears to have bat origin, along with a bunch of other viral pandemics that have been significant: SARS-1, MERS, Ebola, etc. And that has led actually to a lot of interest about what is it about bats that are particularly interesting and make potentially susceptible or make viruses that are particularly dangerous to humans, right? So, there are people who are doing more traditional comparative biology, comparative pathophysiology regarding, bats and humans, looking at some of the specific molecular characteristics associated with bats. And my question that I wanted to look at is there something about the adaptation that bats have to viruses related to their kind of unique physiology that causes the particular disease manifestation when those viruses happen to get into human beings? And I'm thinking primarily of cytokine storm, and this idea of the host response being a primary driver of disease manifestation in acute viral infections, which seems to be the case. All right, so what is it about bats? Well, one of the most startling or the most notable things about bats is that they're the only mammals that fly, that have active flight, and therefore they have a unusually high degree of metabolism, even for other similarly sized mammals. But certainly, when they are flying around, they have an increased metabolic rate of 10 to 30 times greater than similarly sized mammals. And that produces a lot of metabolic stress and DNA damage, because the fact is that cells can only take so much, right? So they have a increased amount of extracellular DNA as a result of the cellular damage associated with high metabolism, and yet they're able to deal with that. They also have certain enhanced features specifically directed against virals, mostly related to their enhanced type 1 interferon properties, and partially related to the consequence of the extracellular DNA and metabolic damage. They have dampened inflammasome activation, which is, again, pretty significant potentially in terms of human pathophysiology of disease. So the goal here is to determine if these differences are sufficient to describe the difference in bats in viral human response to viral infection. So a couple of things about this. And it's related to the model that we came up to investigate this. So the first thing that, if you look at some of the features of this, which I'll go into in a little bit more detail, is this is innate immune stuff, right? So there are no lymphocytes in this current model. It doesn't have to do with adaptive response, cellular memory, any of those sorts of things, which are obviously very important in human disease pathophysiology, but don't seem to intersect as much with respect to what it is that makes bats resistant to viruses. And this again goes back to the the blunting of the response to pathogen and damage associated molecular patterns, which are the byproducts of metabolism. So this particular comparative biology immune ABM focuses on the innate components of the response. and is pretty abstract, as you can probably tell, and is focused primarily on being able to create a minimally sufficient representation of those components to explain the resistance of bats to viral particles. Also, part of the motivation of this particular project is to see if this comparative biology approach can find common pathophysiologic features of a potential zoonotically bat-originating pandemic virus that doesn't require a great deal of detailed knowledge of the specific virus itself. Right, so there's nothing in this particular set of experiments related to the susceptibility of a particular tissue to the virus, the particular binding characteristics of that particular virus, any particular anti-apoptotic characteristics of a particular virus, you know, that type of thing. It is a general examination of what happens in terms of the innate immune response to epithelial infection by virus. And epithelial is chosen because that's very often the primary tissue interface in which viral particles enter mammalian systems. Right, so this is a diagram of the type that is shown very often. You can see the cell types that are present. The epithelial cells are the primary tissue level. Agents, it's an agent-based model instantiated in NetLogo. The inflammatory cells are macrophages, dendritic cells, and NK cells, and neutrophils later on. There are three modes of cell death. There is necrosis, which is a pro-inflammatory cell death. There is apoptosis, which is a non-inflammogenic form of cell death, and pyroptosis, which is a inflammasome-mediated pro-inflammatory type of cell death as well. Interestingly, this model also incorporates endothelium and PMNs, which are not necessarily often explicitly noted in the discussion of viral infections because they usually reflect something further downstream. PMNs generally are not considered to be a major immune cell players in viral effect, but as we'll see, they turn out in this particular circumstance to be surprisingly and potentially important. All right, so this is kind of the overall schematic of the agent-based model. Most people on this call are used to agent-based models, and this type of thing is not super novel. Also on this particular diagram, you'll see a couple of black squares with B numbers in them, B1, 2, 3, and 4. And these represent particular areas, specific areas in the CBIABM that are manifesting the differences between bats and humans. So the idea here is that we are essentially locking down the structure of this particular set of interactions. This is a very common approach in all biology where we use surrogate experimental animals, and we say this system is similar to that system, and therefore we can inform other models in that fashion, and that's what this model is. But rather than focusing on the similarities across particular species, this model is intended to explicitly examine those differences that have been noted by bat researchers as being important features in viral resistance. And those are the generation baseline level of PDAMPs as a result of increased metabolism. Bats have basal constitutive levels of type 1 interferons produced by their epithelial cells. They have enhanced antiviral properties associated with their type 1 interferons in terms of intervening with viral replication, and they also have dampened inflammasome priming. So one of the things, as I made a transition from modeling bacterial infections to viral infections, is that the primary way of the body to control viral infections is by killing infected cells, which is through the induction of apoptosis, as you can see here. Right. So this is kind of interesting because very often there's not direct killing of extracellular virus, at least that I could actually see without preformed antibodies. Most of it has to do with the interruption of cell factories and eventually reducing the reservoirs that are available in order to make viruses. So, yeah, so when I developed the CBIABM, it was initially parameterized to human, right, to create a continuum of tissue damage, initial viral load, and to match cytokine dynamics. So the asterisk there is that, obviously, since it doesn't include the adaptive component, once you start getting past about 5 to 7 days, when you start getting T cell expansion, your cytokines that you get from experimental animals are not applicable. What we do know is that prior to that particular phase, you do start seeing rises and peaks of a lot of these inflammatory cytokines at around 72 hours, and therefore calibration of this particular model was intended to generate those particular dynamics. The other thing that's present or not present in this model is explicit cell steady states. The inflammatory cells are modeled as essentially being immortal, with one particular exception, or two exceptions, which I'll get to later. And the idea here is that within the time frames here, and for the purposes of examining the differences between bats and humans, I didn't want to get into the whole details of modeling precursors, etc. So the populations of macrophages, dendritic cells, and NK cells are assumed to be constant for the duration of these runs. These runs are simulated for 14 days. And again, the reason for that kind of constrained time frame is the idea that the primary features associated with bat differences manifest in that early phase. These differences are seen in the next bullet points, which I've already mentioned, and therefore the aim is to show bat resistance to an equivalent range of initial viral load, right? Because the idea is that if the bat is resistant to viruses, if you give the same amount of inoculum to a bat, it does not kill the bat, whereas it can cause disease in human beings. So here are some cytokine trajectories, and we have two injury numbers that are initial inoculum numbers that are represented here. These are blue, which is the lower initial inoculum number. These are unitless, and therefore the actual value is meaningless. But just to show a difference between disease severity with a relatively mild levels of system damage present with the blue ones, and a higher degree with the red ones. Now, the interesting thing in the red ones isn't necessarily the initial peak, but these kind of late spikes that are present. Again, remembering that there is no adaptive component, but it means that there's some process that's happening with the higher inoculation numbers. Some sort of containment is broken that leads to a second wave of inflammatory cell activation. So as with all these sorts of things, you learn a lot more while you're making the model than with the model that you're necessarily done with. And in this particular circumstance, this is exactly what happened. So I implemented the differences in the cells that the literature focused on, mainly the TNF, type 1 interferon manifesting epithelial cells, and the emphasis on inflammasome activity in macrophages. And, I thought this is what people think is going to happen, and therefore this will be cool. But it turns out that didn't work, because when I did that, I was unable to get significant separation in outcomes with the metabolic byproduct variable, which is that metabolic stress from increased metabolism. Once I got much more, about twofold higher in bats versus humans, the amount of system damage that those simulation runs had were very similar to the humans. And that means that for whatever reason, that particular hypothesis structure was insufficient to explain the resistance of bats to higher viral loads and disease resistance. It's supposed to be at least 10 times with essentially no disease manifestation. So there's a problem here. And of course, I wonder what's going on in this particular circumstance. So then what I did is I looked at those late spikes and just watch the simulations run. And what it turns out is that the endothelium has an inflammasome too. Right. So what happens is apparently, at least within the model, and maybe what happens in the real world, is that at some point in time, the tissue inflammation that's being generated is enough to activate the endothelium adjacent to that epithelial tissue, and therefore lead to recruitment of systemic factors. And this is essentially system level spillover of whatever is going on with respect to the disease process. And that is the acceleration or the conversion of what is otherwise a local disease to a systemic manifestation, which leads to the more severe disease manifestations. In the initial simulations, I assumed that these were similar between bats and humans, but it turns out that if the endothelium has an inflammasome and that inflammasome has reduced sensing in the macrophages, it probably has reduced sensing in endothelial cells as well. Now, the endothelial inflammasome, as opposed to producing cytokines, is linked to the expression of adhesion factors that are related to neutrophil recruitment, and therefore, I assumed that there's an increased threshold in bats. It turns out there's no literature on this. Right? I looked to find about whether or not people look at this role in bats, and it turns out not to be the case. It is present in the human disease pathophysiology, reflected as the coagulopathies that people talk about. So having done that, now the system works. If you assume the same types of inflammasome dampening that are present in macrophages are present in the endothelium in bats, then you are able to get significant separation in the relative levels of or the relative responsiveness of bats versus humans. So what we see here, and this requires a little bit of explanation. So what we did is we ran 1,000 stochastic replicates at each initial inoculation number, right, with increments of 25. And what we see here, so instead of plotting like 1,000 different bars, basically it's being plotted as a line, and essentially it's a distribution of all the outcomes of all these runs, 1,000 runs, with runs that did well on this side and runs that didn't do so well that way. And it's ranked so you have a smooth curve. So the area under the curve actually represents the aggregate of the system death for that particular population. So as you increase the amount of inoculum, the human does worse, as you can see here. But if you do that across the range for the bat, it essentially stays in this range up here. And they're very resistant even to fairly high initial inoculum doses, right? And you can see that there is at least some degree of scaling or some dose responsiveness in the bat number. And this is a magnified view of what's happening in that little screen. So this is a that says that the model reproduces what we think plausibly the differential phenotypes are between humans and bats. So that's all kind of good. So since I came up with this idea that the endothelium is important, we wanted to kind of test this and see, well, let's see if we can make the human more like a bat and see how far we can get in terms of doing that. So what we did here is we held all the human parameterizations the same, so they don't make interferon. Constitutively, they have decreased or increased macrophage inflammasome activation, and they also have they don't have the enhanced antiviral effect of type 1 interferons, but we are increasing the resistance of the endothelium to activation. And what we see here is that as you increase that level towards the bat, the bat is at 10, right? The human is 10 here. It's not exactly the same. So those other features of the bat are still protective at some degree, but it is substantially better, right? For what otherwise would be a fairly significantly dangerous level of viral load.
Right. 5 minutes.
Okay. So that at some level points to a potential therapeutic target, because if the endothelial activation happens to be the expander of the process that leads to the manifestation of disease, that might be something to investigate as a more generalizable disease mitigation pharmaceutical target. One other thing that people are interested in is what causes the zoonotic spillover in bats and increased metabolic stress beyond what their baseline metabolic stress has been invoked as a reason for that. So, ecological stress, hibernation changes, etc. So what we did was we decided to increase the metabolic stress of the bat, this MB term, and see whether or not we could make the bat sicker. And it turns out you can, but interestingly enough, it does seem to have a lower limit here for how sick you can actually make the bats. This is again preliminary as much as the other work is, but is interesting. So insights and future directions for the CBIABM, right? So as with a lot of these modeling experiences, as all of you know, you gain a lot of insight just for doing the model. So this model insufficiency based on the initial approximation suggested this potential novel role of endothelial and inflammasome. And we're talking with some collaborators to see if we can do experiments about this. And as I alluded to, a potential therapeutic target. The limitations are almost too many to enumerate. Obviously, this is a very abstract preliminary model. There are a whole series of simplifying assumptions that are made. I think one of the things to keep in mind, however, is that with the intended purpose of this model, which is to examine the specific differences between humans and bats, I think it's pretty good because the general dynamics associated with those processes are manifested in the features of this model. But these other things obviously will be added as we move forward. And future directions are to add adaptive immunity, as alluded to before. We have this particular process to do extensive parameter space characterization using genetic algorithms and active learning. The model is too primitive at the moment to do that with. All the stuff that I showed you is essentially hand fitting of the model at this point. But again, it just shows a sets of plausible or interesting outcomes, which spur other questions, which is to a certain degree the whole point. And of obviously virus-specific features. So any sort of endothelial targeting therapy wouldn't necessarily help if the virus necessarily infects endothelial tissue, which COVID happens to do. But again, the idea here is whether or not we can get upstream of the next pandemic and try and find a kind of a potential therapeutic target that isn't necessarily specifically dependent upon knowing the particular pathophysiologic features of a specific viral species. So with that, I'm done. I'll take any questions and looking forward to your comments. Should I stop sharing?
I think in the interest of time, we'll do the second talk and then we'll have the questions, if that's
Great.
okay.
Yep.
Okay, thank you very much for the great talk, Gary. That was impressive and a lot to think about in that work. And now we'll move on to our second speaker, Keisuke Ejima from Indiana University. And I will let him take it away.
Thank you for kind introduction. Let me share my screen first. Can you see my screen now? Oh no. Can you see my, no? What's going on? This is weird. Can you see my screen?
It shared, when you shared it the first time, it came up
Okay.
and then disappeared. So maybe unshare and reshare. There you go.
Can you see the screen?
It says it's launching.
Okay. Mhm.
No, it dropped again.
Sorry about that.
It's okay.
So how about this?
We can see your slides. It's in the regular, it's not in presentation mode, but that's fine.
I think, yeah, I will do with this. So,
That's
my
fine.
name is,
No worries.
thank you for kind introduction and thank you for this opportunity to speak about my current research on SARS-CoV-2, COVID-19. I'm an assistant research scientist at Indiana University School of Public Health, Bloomington. And before jump into the research topic, I just want to share my background. My background is mathematical engineering, and my interest is not just building a model and analyze data, but also I'm more interested in the application of the analysis using model and apply the modeling and analysis to solve the issue in public health. So in terms of the SARS-CoV-2 research, I generally use a viral dynamics model that describes how the virus replicate themselves. Using the model, we analyze the data of viral load and apply the analytical results to solve more practical issues, for example, estimating an incubation period or predicting the antiviral effect and so on. So there are a lot of study ongoing and or published already, and basically all of them are modeling or methodological perspectives. It is very simple, but the uniqueness of my research is, I think, the modeling approach is used for various public health issues. So here's some example. One is I applied the modeling approach to solve the issue in epidemiology. And the second one is assessment of test quality, actually PCR test quality. And the third one is epidemic control. And the fourth application is clinical trial design. But in terms of the time, I think I will focus on the first three topics. So I don't want to go into the detail of the model because we don't have much time, but I'm happy to discuss after this presentation. But basically, the mathematical model I use is very simple, composed of only three variables: uninfected cell, type infected cell, and virus population. So uninfected cell turn to be infected cell depending on the date. This rate, beta V, V is a virus population. And infected cells die by the rate of delta, and infected cell produce viruses by the rate of P. And the virus, of course, produce infected cell, but also it dies by the rate of C. So here is a very simple model, ODE model. And actually, we can use a QSS assumption to reduce the dimension because the death rate of the virus is much, much higher than the death rate of infected cell. So we can reduce the dimension of the model. So here is a kind of reduced model. So in this model, F is the defined as here. So the proportion of target uninfected cell at time t compared against the uninfected cell at time 0. And this is a very simple model, but there are some interpretable quantities derived from this model. So one is, for example, the duration of virus production, 1 over delta. So again, the delta is the death rate of infected cells. So 1 over delta is a mean length of the infected cell lifetime. So that is considered to be duration of virus production. And the second one is the basic reproduction number. This is kind of similar to the reproduction number in the infectious disease model. So it can be considered as the number of virus produced by a single virus. That is calculated as gamma over delta. And the last one is critical inhibition level, which is the proportion of uninfected cells. How can I say? It's, the idea is similar to the herd immunity in the infectious disease epidemiology. So in that case, there is a critical vaccination level. So if some portion of the susceptible population is infected, the virus never spread among human population. So the same idea in this model, if the disproportion of uninfected cells are protected from infection, then the virus doesn't propagate, replicate themselves efficiently. So this is a model, and we parameterize the model using the viral load data of SARS-CoV-2. To parameterize model, we need a longitudinal data, and actually it is challenging to collect the longitudinal data from the literature. So far, we have access to 4 papers, and the data are in total 30 patients of the data are available. Especially because the reason the available data is limited is we want to make all the data kind of consistent in terms of the type of the specimen. So in these 4 papers, the viral load was measured from upper respiratory specimens. There are many, basically, I think that collecting the upper respiratory specimen for measuring viral load is a common approach, but still the data are still limited. So we fit the model to this data using mixed effect model. So the reason we use the mixed effect model is, again, the data are limited. So for example, for some patient, the data points are more than 10 data points are available. But for some patient, 4 data points or less are available. So we want to estimate the parameter for each individual, but it is not feasible without using the other data from other patients. So we use this modeling approach. So we estimate not just a fixed effect, which is constant across patient. We also estimated random effect, which is different between patients. So we can reasonably estimate the parameter for each patient, even though the data are limited. So here is an example. the dot are the data, and the line show the fitted curve. So for example, this person, Singapore 18, the data are available a lot. So, but for this person, China D, only 4 data points are available. But we still can fit the model because, again, we estimate not just a random effect, but also we estimate the population, fixed effect parameter. And interestingly, there are kind of variability in the dynamics. So for example, this person, the duration of viral shedding is kind of long, over actually 25 days or something like that. But for China D, the duration of viral shedding is less than 20. So there is a huge variability in the viral load dynamics. So using this model and fitted parameters, what we can do is, we can do out of stuff. But for example, in the epidemiology field, we can estimate incubation period. So the question is, how long is the time interval between infection to onset, which is called incubation period, the time interval between infection to onset. So again, we fit the data. So this is a fitted curve. So if we back calculate the equation after fitting the model, we can estimate the timing of infection for each individual. So this purple area show the timing of infection. So for this person, it seems he has infected about 4 days before the symptom onset. And for this person, he or she seems to be infected before 6 days or something before the symptom onset. So we estimated that, so this is the incubation period. So we estimated the incubation period for all the patients, and then fitted the log normal distribution for this estimated data. And so this is the incubation period distribution. When I use all countries' data, the incubation period distribution becomes like this. And if we only use Asian data, the distribution is like this. The same thing for European data. But even though I use Asian data or European data or all data, we constantly find that the median incubation period is about 5 days, which is actually consistent with the previous studies. So the thing is, we could estimate incubation period using the viral load data. Actually, usually the incubation period is estimated based on the contact trace surveillance. That means the timing of infection is inferred by interview based. So it's self-report, so it's basically very biased. But using this approach, we can maybe combining with traditional approach, we can accurately estimate the timing of infection and also incubation period as well. And this is a second application. The question is, what is the probability of PCR test negative given that one is infected, which is called false negative rate? So here is a summary of the computing process of false negative rate. So again, we fit the model to the data and obtain the parameter distribution of the model. And these run simulation based on the sample parameter set. Then we assume the obtained viral load, the viral load curve as true viral load, and we add error term to the true viral load. the error is assumed to be follow IID assumption. And we perform sensitivity analysis varying the detection limit. So it depends on the detection limit, but here is the false negative rate over time. The time scale is days after symptom onset. So what we can say here is around the symptom onset, the false negative rate is very low. It's very close to zero. That is because of the high viral load near the symptom onset. But the problem is if the test is performed before the symptom onset. So if the test is performed, for example, 2 days before the symptom onset, false negative rate is over 50%, 60%. So this is problematic. If we perform, we should not perform we can diagnose the infection if the test is performed before the symptom onset. And the false negative rate is dependent on the detection limit, but we can see a similar curve, even for the detection limit is 100 copies per milliliter or 1,000 copies per milliliter. The reason we use this approach, the modeling approach, is that the data are based. There are a lot of studies investigating the false negative rate over time, over the time course of infection. But most of the data are available after the symptom onset, because that's not really surprising, because because most of the people visit the hospital and get tested after symptom onset. That means there's not much data before the symptom onset. But using this model, we can estimate the false negative rate even before the symptom onset. That's the strength of this study. And third application is the model can be used to control epidemics. So the question is here. In the early phase of the epidemic, one patient was diagnosed by COVID-19, but was the patient infected in or outside of the country? Why this is important? So here's a schematic illustration of the identify the patient. So let's assume this person is infected in country X and traveled to United States. This person infected in this country, but given the symptom before the departure of the country X, then he can get on the board. That means he will not be able to go to US. But this person cannot go enter the United States because he developed symptom in the flight. But the problem is this person. He got infected by the virus in country X, but developed a symptom after the arrival to United States. So he may transmit the virus to the other people. So this person is considered to be important, and these people are considered to be local infection. And from the epidemiological perspective, it is really important to distinguish between these two, imported cases or local transmission. Because imagine that it is in the early phase of the pandemic. If most of the cases are imported cases, then we can still do a containment measure because the local infection is still limited. But if most of the cases are result of local transmission, then that is a problem. We need to switch the strategy from the containment measure to mitigation measure, because we have to control the local infection rather than stopping the importation of the virus. So that's also possible with the virus dynamics model. Again, once we fit the model to the data, we can estimate the timing of infection. So we applied this approach to the Singaporean data. This is actually, there are 12 cases considered to be imported at very early phase of the pandemic. So we fit the model to the viral load data of each person. So we could estimate the timing of infection, which is represented by blue shaded area here. And this red dotted line showed the timing of entrance to the country, Singapore. So what this means is the timing of entrance is after the infection. That means this person is imported cases. And the same thing for this person and this person. But we couldn't control that these three persons are local cases or imported cases.
Okay, just 5 minutes warning.
Okay. Thank you. So these three persons could be local cases. And the same thing for the other 6 cases. These 3 seem to be imported, and these 3 seem to be a result of local infection. So to validate this approach, we used this data. This data was collected after the, how can I say, this is a result of contact tracing. So the number of case ID corresponds to these ones. So these people with blue circle is considered to be imported from our model. And these people with red circle is considered to be local infection from our model. And interestingly, if we focus on this person, the patient ID is 18. This person is considered to be local cases from our model. And actually, this person is connected to this person. So it seems to be this person was infected by this person. Initially, all the patients are considered to be imported cases, but that's not really true because there was a connection between these two. And the same thing for this person and this person. So we can, I don't think we validate the model 100%, modeling approach 100%, but seems to be working, somehow working to identify some of the imported cases, local cases using our approach. So here future work. Again, it is challenging to collect longitudinal viral load data. Actually, the data we use is, again, here is collected at the very early phase of the epidemic. This is because people are more interested in the viral load data from the clinical perspective. But now, I don't know, but I think the researchers are losing their interest in terms of collecting viral load longitudinally because maybe it's not really useful from the clinical perspective anymore. But obviously, there are many variants like Delta variant and other variants. And also, I think there should be variability because of the age, sex. So we need more data, especially if these variables, these factors are associated, these factors changing the viral load dynamics. And again, the model we used is very simple. So we need to make the model more biologically sound. So for example, we didn't consider immune response clearly. So we might need to update the model, but given that if we need to the model and analyze the data are very early phase of the epidemic. We don't know the detail of the dynamics. So I think using the model, using simple model is reasonable. But again, now we know more about the immune response and so on. So we need to update the model. And I'm also interested in the further application of this model in the context of epidemiology or public health. So those are ongoing work. And these are my collaborators from around the world, not just United States, but also Japan and UK. So thank you.
Okay, thank you very much. That was great to see the connection between an in-host model and an epidemiological model. Because one of the things that we've wanted to do very much is to be able to connect between scales. And so it's very interesting to see that kind of application. So our canonical 4 o'clock has arrived, so it's time for discussion. So we are open to questions from anybody, either of our speakers. As usual, I have both breakout rooms open if it seems that we need to use them, but I think the topics are relevant to each other, so probably we can talk as a group. Well, I guess I have a question for Keisuke to begin with. You mentioned that the availability of time series data got worse as the epidemic went on. Yeah, that's a bit surprising to me. I know one of the problems that people like doing this kind of modeling, like Alan Perlson always face, is that you don't get the leading edge of the viral load because you've reached the maximum before people feel sick. So you have to have some kind of prospective monitoring of viral load in order to be able to do it. What could you tell us if you had an ideal viral load curve? What would it look like? What data sets should we be collecting?
Well, one more thing in my mind is, for example, the Olympic Games is going in Tokyo, and they are doing a PCR test every day, even though those are symptomatic and asymptomatic. So that is, I think, the great opportunity to collect the data before the infection actually, so we can capture the dynamics in the upper slope and lower slope. So that should be a great opportunity, but I'm not sure they share the data or systematically collecting data. But yeah, again, we need to collect the data before symptom onset, if possible.
Lorenzo, question.
Hi, thank you both for the talk. They were both amazing. I have a question for Gary. You mentioned endothelium. It happens that I'm very much focused on studying endothelial cell in vitro. I was wondering, it was saying, all right, so level of endothelial inflammation might be one key to understand more or less damage that the virus can do. Can it be, in case of humans, since the endothelial cell might be getting infected, a form of reservoir, or might have an effect in long COVID, on the long-term effect of COVID? I'm thinking also about shedding the virus in different endothelial cells than those in the lung. Do you have any ideas, or have you put some thoughts there?
Well, so I think, you know, the specifics of SARS-CoV-2 versus kind of the hypothesis that I proposed, I think the distinction that needs to be made is that I think it's pretty well recognized that endothelial cells have the receptors that allow infection of the virus per se. And I think when you're dealing with that particular circumstance, it's a little bit different because now you're dealing with having to contain the virus and then dealing with those as opposed to induction of its inflammasome through collateral host effects, right? So certainly the let's call it coagulopathy, endotheliopathy associated with COVID-19 is clinically well recognized and is currently a very hot topic of therapeutic investigation. The thing that I found interesting is that you don't necessarily have to invoke endothelial infectivity with that before getting disease manifestation associated with a viral infection. So, all the issues you talk about are fantastic. I think that, specific to COVID-19, these are very relevant issues that need to be discussed. I think the thing that I was interested in coming out of this particular study is a early disease mitigation strategy. So, for instance, for the 1 year it takes in order to get vaccines online, during the time in which you're trying to determine whether or not effectively potentially repurposed drugs are going to be, effective in the particular circumstance of a particular type of viral infection. Are there any sort of like pathophysiologic choke points that can potentially be targeted that might be a generalizable disease mitigation strategy? And I'm not saying that this is necessarily, the answer, but it's not, I mean, people haven't studied this very much. So I think that, if you are interested in studying the endothelial inflammasome activation with me, I think that would be great. One of the, experiment. I mean, so here's a very low bar experimental process, experiment you could do, where you take a known, fairly significant non-COVID viral disease, right? That doesn't affect the endothelium, but manifests systemically, like, flu or something like that. And you just mitigate pulmonary endothelial activation through whatever you use, selective knockout, some pharmacologic silencing, whatever, right? That you attenuate endothelial inflammasome activation relatively early, right? And then or early with respect to when you would expect viral incubation to occur, because the idea is that you are trying to prevent the breaking of the containment from what is otherwise a manageable local tissue infection to disease severity. Now, it doesn't address transmission. It doesn't, address a whole bunch of other things with respect to the containment of the pandemic. But in my opinion, a significant public health impact of the pandemic is that it made people sick enough to consume hospital and ICU resources. If you could take it, and this is what the vaccines are doing, it's taking people who get the disease and making them non-hospitalized people. I mean, that is really important to be able to do while you're bringing these other things online. So, if that's a set of experiments that you'd be interested in doing, then we should talk, because that would be the exact sort of question raised by a model, experimental investigation, refinement, whatever cycle that, that we hope these modeling projects do.
Thank you.
Okay. I guess let's do John and then Ruchiro.
I think Ruchiro had her hand up first. Why
Oh,
don't
I'm
you-
sorry. I saw. Okay, sorry,
I
Ruchiro.
actually thought John did, but anyway, we can. Anyway, so I don't mind going. So yeah, this is really interesting, and thank you for sharing work at this stage. So it seems like it was handy that you could get some reject a hypothesis out of the model and then add something, and then that suggests that that's what is a possible driver. So as you might, I don't remember if you were there when I discussed approximate Bayesian computation for agent-based models, And one of the things it can do is, if you investigate the parameter space, and you can sort of design ways of rejecting hypotheses. So, like I mean, you only had a few minutes for your talk, but how have you been doing that? Like was it just sort of like empirical, or how were you determining that, this did work or this didn't work? Or
Yeah, I mean, that's a great question. So this is all hand fit, empirical, standard hacking things together at the moment. There's not a systematic parameter space exploration done with this particular model. I do think, however, that having stumbled upon, at least within a particular range, a pretty broad range of implausibility, because it's not like I didn't try to make it work, right, within the constraints of not having to start invoking some feature that wasn't there. I can't say that it was comprehensively explored. I can say that I spent a lot of time trying to figure out what the heck was going on and why this wasn't working appropriately, and trying different combinations. And I think the thing which a lot of us recognize is that the dynamic range of behaviors of these models is actually pretty constrained, right? I mean, I think that there are fairly defined boundary conditions with respect to what the parameters can or cannot do. You don't necessarily need a huge exploration of that entire parameter space to identify when the thing is not working correctly. I think identifying failure points at the edges of that parameter space is more important. But I think if in the process of just trying to do a hand fit and it not working, when you start with what appear to be pretty plausible parameter values, if you can't even get it to work at the beginning, then I think there's probably something wrong with the model structure.
Right. Yeah, no, I'm not saying that. I mean, yes, I agree that that you did find that the model structure had to be modified. I was just suggesting that that might be a way of well, I guess it depends on your aim. Like if you wanted to say write a paper and explain that, justify it, then that could be a justification. If you're just trying to find the correct, sort of iterate quickly and find the correct model structure, then it's probably overkill.
Yeah, I think the model structure is recognized to be insufficient enough, right? So that we're going to find areas in which it breaks pretty readily. I think when we get to something that has a higher degree of biological realism, that's trying to fit to some actual data sets, then like the approach that you're talking about and some of the other tools that we use to explore parameter space, those things then become more relevant. I think at this point of kind of, early model exploration, it's not necessary. In fact, so the original intent of this was just to show plausibility, and this additional finding is something that came as a surprise during the development process.
Yeah, that's pretty cool.
Yeah, I'm wrong often early. So yeah.
Well, they say failure is the pillar of success.
There you go. I'm good at that, failing at least.
Okay, John.
Yeah, I had also a couple of questions for Gary. Very interesting talk, and these are more showing my ignorance rather than anything else. One is a very broad question, and that is whether I'm imagining that there are viruses that do infect bats and cause severe disease, and I'm wondering if what's known about them and how they might differ from the SARS, from coronaviruses.
Yeah, so I mean, I think that's a great question. The short answer is I don't know. I mean, I've pretty much exhausted my knowledge of bats. I've just read what review papers are there and the references that are in it. So such things certainly do exist. I think one of the things that is interesting is that you might presume that bats may be more susceptible to other types of pathogens that rely on a more aggressive innate response, and that seems to be true. So like there are a whole series of fungal infections and otherwise potentially containable types of infections that rely more on effective innate immunity to clear that they seem to be susceptible to. So I think, there's an entire world of comparative pathophysiology that can be investigated. At some level, if viruses are killing bats, then the bats are probably not going to be particularly large reservoirs of that particular virus in terms of human health.
Yeah.
But I think it is an interesting sort of thing to see what
Yeah.
the invasion strategies are of those particular viruses.
Okay. And the second question was more on the your setting up of these agent-based models. I think you mentioned that they were parameterized based on humans, and I guess human and kinetic data on the signaling pathways. Can you say a little bit about how that data informs the time step that you use, and whether or not you encounter situations with separation of timescales when you do these sorts of simulations?
Yeah. So the way I do this, which is distinct from using standard kinetic rate constants that are present in a lot of experiments, is because there's no master equation, right? It's a series of essentially different arithmetic equations that iterate over time. So, there's just kind of an update associated with that. And it's usually arithmetic as opposed to multiplying, dividing, because then you can run into these kind of exponential problems, et cetera. So there's just a lot of fiddling around, right, to get these constants to generate time series spikes that appear to be reported in the data, in the literature. And I think, all of us are very aware of what the potential confounding factors are with respect to experimental data and what those are. So there's like a general understanding that these cytokines have a tendency to peak within this general time frame, and that's kind of as detailed as we're trying to get with respect to the initial hand fits, right? Because we're just trying to see whether or not we're even within the reasonable ballpark. And again, in this particular circumstance, it's just to show that the general properties of a responsiveness to the viral infection, some degree of containment and potential resolution is possible with this particular system that follows the general dynamics of what is observed, and then keeping most of those features constant in doing the comparison between the bat and the human. So it's entirely possible, and it's almost certainly true, that there are going to be variations in some exact specific kinetic constants with respect to cytokine, degradation and production between bats and humans. That's almost certainly to be the case. The question is whether or not you're going to get this degree of qualitative difference in behavior as a result of those particular parameters. My supposition is not, but you would have to do a much more comprehensive parameter space exploration of the type Richard was talking about, and we've done for other types of situations in order to completely answer that question.
So in short, it's essentially a single time scale associated with a time scale of when the peak occurs. And I guess if you thought about longer time scales, they might be processes that would be a priming from an initial infection that somehow sets you up for protection on longer time scales.
Yeah, so the way they are, at least ABMs I make work, is that they iterate based on a particular time step per run. So it's 10 minutes for this particular thing. So the time scales that are present for longer phenomena would emerge out of those particular dynamics. So when we get to the point where we're modeling long-term immunity, right, we're going to have to run simulations out to 6 months or so at 10-minute increments. So we may at some point in time, having run a couple of those, use proxy systems like artificial neural networks or equation approximations, et cetera, so that we don't actually have to step every 10 minutes during a time when we don't think something is actually happening, actively happening. But it's a very a naive approach towards dealing with longer time scales. We just run the model longer in order to get that. Yeah. So, for instance, the sepsis models that we do, we run out to 96 days of simulated time at 6-minute increments. So they just run a lot.
Great. Thanks a lot. I appreciate your insightful comments.
Thanks for your question.
Okay, other questions? Luis?
Hi, Gary.
Hi.
Great presentation.
Thank you.
What you said, so in another life, I worked with malaria, and some of the things that you said are very similar, although malaria, no, it's not a virus, it's a eukaryotic parasite. But when you infect a primate with malaria, the parasite goes through a few days in the liver, where it's supposed to be asymptomatic, but we have detected some form of inflammation there. What I just wanted to say is, if you ever, so you were saying something about not having a lot of data. I believe we would have data in most of the nodes that you showed on your network. We would have time series data for most of those things available if you ever wanted to try malaria.
For malaria. Yeah, so that's very interesting. I was at a malaria vaccine conference a couple of years ago at the NIH, and we introduced the idea of potentially using mathematical modeling for vaccine, development. And of course, I remember from medical school just how complicated the life cycle of malaria happens to be. So that is that's a huge endeavor. I know there are excellent groups who are doing that kind of work. And if I want to move in that direction, I will certainly let you know.
But we have public data that would cover all of your nodes.
Okay, that's great. Thanks.
Ruchira again, and then John.
Oh, sorry. I hadn't meant to ask the question, but I guess I had left my hand raised. But since I'm called, I have been reading recently that, and I guess this is a question for Keisuke. I just read recently that in the Delta variant, the viral load is much higher, much earlier than with the Alpha variant. Like there's like thousands of times. So is that being determined in the same way as, or is that a hypothesis? How is that being determined?
Actually, I think the data are from more like cross-sectional data rather than longitudinal data. And yeah, I think so.
Okay, thanks.
Yeah, but to fitting the model to the data, we need a longitudinal data because we're fitting a kind of model to individual data from individual patients. So.
Right. So it's something that people often do is they just measure across a population and then infer something about. But it's kind of like the difference between single cell and bulk.
Mm-hmm.
Yeah. Okay, got it.
Okay, John.
Yeah, a couple of things. Part of it's a little bit of background, but early in the pandemic break, when I started asking a lot of questions about bats and why we die and they fly, and I actually went to James and Gary, and we had several meetings with a bunch of bat scientists. The last of which there were 12 bat scientists and the three of us. And when this group was organized, one of the proposed subgroups was zoonotic modeling. That was partly out of that in the bat community, when James or Gary would talk about multi-scale modeling of bat immunology, the multi-scale concept was not well known in the bat science world. And so that was the reason a zoonotic subgroup was proposed for this working group, so that we could exchange information with them. And I would still like to suggest that I mean, apparently nobody who joined the virus multi-scale working group had bandwidth to talk about animals. But if there are other people or any of you that are interested, certainly at a minimum to schedule a couple of those bat science people for presentations at our meetings here. And at the time, there were lots of questions. You know, the one that Gary was addressing dealt with the infectious process itself. One of the other questions what would understanding the host organism help tell us about the virus and the potential before the virus mutation even showed up? Among the interesting things were that when the bats are hanging around the cave, their body temperature is 97 degrees. And when the bats fly, their body temperature goes to 107 degrees. Well, if the bat's body temperature goes to 107 degrees for 2 hours twice a day, what does that tell us about the resistance of any surviving coronavirus? It says coronaviruses do not succumb to human fever before the human would succumb to the human fever that would take to damage the virus. So there's a lot of questions in both directions. I think that that I can't find any community where the bat experts and the human multi-scale modeling experts have come together. So if there's any interest, I would love to try to reinitiate that.
Yeah, so John's part of the motivation of doing this work and getting an early version of the work out was just as a proof of concept for some sort of potential benefit of this type of representation, because I'm working on the paper regard to this and looked at the number of existing in-host models of bat response to viremia, and it's really very, very sparse. There are not very many, and there's only one that has any degree of any molecular detail. Most of them are essentially susceptibility, infection, resistance models that aggregate all these properties and just model some of these issues and the bat community is very interested in what causes spillover, right? Because they are curious about the ecological features that would lead to the potential zoonotic jump from bats to humans. So I think, I'm certainly interested in that. I think one of the challenges we had when we were talking with them, just, to fill everyone else in on James and my own conversation with them, was giving them something as a proof of concept. I think I proposed trying to do what I did with this model with our old, with our 20-year-old innate immune model. It just didn't work because you couldn't simulate viral infections with that model, and therefore had to come up with this new model to be able to simulate viral infections. And therefore, the hope is that maybe we can get some of that conversation going. As we all know, trying to get experimentalists on board with the benefits of this type of modeling has been a challenge in the medical side. It is a similar challenge on the bat zoology type of side. I mean, they have viral transmission dynamic type of models and stuff like that, but they are these kind of aggregated bulk entity types of things.
There's just so many curious things. It's like the infection, COVID is a respiratory infection, and people get it and they can't breathe, and we have to assume that for that bat to fly, the oxygen demand is such that whatever COVID viruses do in bats cannot be interfering with the respiration system. It's just whatever's happening in there. If the bat can't pull that oxygen, they're not going to be able to eat.
Well, I mean, they don't get sick at all from these things. So
Yeah.
you
I
know,
mean,
the
it's
lungs
just-
are not involved. So I
Yep.
think, you know, it may be too far of a reach for NIH or you know, or NSF, but I think if we can get a group together and get, you know, this
Well-
is something to write up to go to DARPA or something like that, because this is
Hopefully, if we can attract a little more interest on the part of NIAID, where the real studies in infectious disease money are, and lots of it, that these might be things that would be more sellable in a NIAID, National Science Foundation collaboration than we were seeing in a biomedical imaging and engineering institute. Okay,
I mean, I think as Lewis had mentioned,
thank
you know,
you
there
for your
are
persistence, Jerry, and working on this stuff.
there are a bunch of vector animal modelers in NIAID, right? Because a lot of these infectious diseases have vector biology that's involved in how those diseases are transmitted. So I know, you know, certainly in malaria, leishmaniasis, I think, is another one that there are, you know, animal vector models. I'm not aware of a bat modeling community in NIAID, but I think that's, again, something that we could potentially pursue, given the relevance of potentially pandemic viruses of bat origin.
I see the time is up, but I wonder if I could chime in with a question that was motivated by John's thoughtful remarks. So the metabolism aspect being heightened for the bats raises the image in my mind of this scaling of metabolism with body mass that you might be familiar with, allometric, you know, where I don't know, quarter power scaling or something of the metabolism with body mass from mice to elephants or mice to whales, right? So something that we found by looking at a lot of virus data was a kind of similarity in time scales between the delay time of a virus. You know, you infect the cell and then it takes a certain amount of time for the virus progeny to come out of the cell. So that's a single virus, a time scale associated with virus production. That time scale turns out to be just about the same time scale as the doubling time of the host cell. At least these are for studies in the lab. So I wonder if anyone is aware of any kinds of studies that do some kind of cross-species analysis of immune response. It could be time scales of the immune response, or is it always on the sorts of peak times that you maybe see for the bat response, or is there a lot of variation? Are there orders of magnitude difference among cells from different animal sources? And it's hard to do this in the lab because everyone wants to use transformed cell lines where oftentimes, we have no innate immune response or something's deficient there. I'd be curious if anyone knows anything that's been done on kind of scaling in immune responses.
I'm not specifically aware of mathematical modeling done for that, but I do know that mice are like 1,000 times more resistant to endotoxin than human beings are, and I think other rodents are. So I can see where the scaling relates to metabolism because, you got to keep yourself up to a certain degree. I think my very naive impression with respect to differential immune responsiveness has been a hard trade-off between self-destruction and a degree of environmental insults that you face. So rodents are getting down in the dirt and scuffing each other up and being around bugs and stuff like that a lot. So I would expect that they would have to have fairly vigorous innate immune responses so that they just don't die from a scratch or something like that. And their life scale, their lifespan is not long enough for them to, from an evolutionary standpoint, worry about autoimmune diseases causing them to not be able to age enough to reproductive, capability. So I think that it's probably not a body mass scaling issue, but I think it is something to do with other features of their environment. So, for instance, these naked mole rats that seem to live forever and have these incredibly robust immune systems. Like, what's about that? I mean, they burrow around in the dirt. There must be something about that environment that causes them to adapt to it. So I think this whole area of comparative zoology or comparative pathophysiology is important. I think
it may not be AI, but some grand challenges around comparative immunology and the application of modeling to do that. If there's people that got the bandwidth to form a group and talk some more about it.
So there are disease-specific and animal-specific things. So like when they came out, the fact that elephants have like 5 different P53 systems or something like that, that keeps them from not getting cancer. People are interested in these types of adaptations with respect to how they impact human disease. I think finding specific circumstances where those attributes are transmissible, right, from a animal environment in which it has evolved to be that way to some sort of intervention that is implementable in the human to affect human health. I think that is the challenge. So it's nice to know that elephants have 5 different types of P53 and fixed DNA damage over their entire length span and orders of magnitude more total number of somatic cells than humans, but you can't give humans more P53. You can't do that. So I think that there are, interesting things. The transferability can't necessarily be directly seen at the first, but maybe getting downstream somehow can do that.
Great, thanks a lot.
Gary, we tried to get funding to actually put extra copies of P53 in mice. Precisely because, and we could never get it done. We could never do it because of Carlo's study on the elephants. Yeah. And we could never get it. They never get any traction on it. I mean, if there were going to be one, if there were going to be one human genetic engineering stem cell genomic genetic, germline genetic engineering that people would allow, it probably would be that. So
unintended consequences, though.
Well, you don't know, but that's why you want to do it in mice.
Yeah,
because you want to know if it depends on how the level of the protein is regulated. If you have 2 copies of the gene, if the regulation is very tight, then the number of replicates doesn't matter, and then you're fine. If it's not, you're in trouble. So that would be, and I don't know if anybody's done that experiment yet.
That's really interesting. I was in Carlos' lab. Aaliyah, his graduate student, was the one who. So yeah, I didn't. That's a sort of an interesting follow-up that you were trying to actually, take it to the next step. Like, okay, let's act on this by becoming more elephant-like.
Right. See if von Neumann can fit that particular phenotype with 3 equations. So.
Well, I guess we're out of time. Unfortunately, I wish there were more time. Keisuke, I'm sorry, sometimes there are questions more for one person than the other. But thank you to both of our speakers for very stimulating talks. And I hope we have the opportunity. Of course, Gary is around all the time, so he could come back and talk anytime. Keisuke, you're always welcome to come and give another talk. You give a talk on the clinical trial design. We'd love to hear about that. And thank you to everybody for staying here. And it's obviously a very stimulating pair of talks. So appreciate your being here. Look forward to talking next week. And I'll bring the meeting to a close.